Good evening class. In last video we have discussed the adject law of dominance, law of aggregations and law of independent assortment, and all the three laws has been given by Mendel's by proving their own inheritance character from one generation to another generation. Okay, but today we are going to discuss some more hereditary characters because we have to study in 10th class the hereditary and evolutions by in a two ways. Okay, here we have to discuss that hereditary and evolution we have to study in a two phase. In first phase we have to study only hereditary. In second phase, in next video, we have to start the evolutions. Okay, now today the class is totally based on hereditary, so we have to follow that. After the completion of all the three laws, the Mendel's has given how the characters and how the traits are transferred from one generation to another generation and which type of changes are generally occurs. Okay, so apart from that, we have to also discuss that. Today we are going to discuss that how these traits means special type of characters and get expressed, and they are also transferred from one generation to another generations. Okay, so we have to start from the first, and that is important part is that cellular DNA is the information source. As we know that DNA is the stand for deoxyribonucleic acid. It is most important part which is found in our chromosomes. Okay, most of the time the question asks that where the exact location of DNA. So DNA is found in chromosomes. Okay, chromosomes you already know that, and chromosomes is found in nucleus. Okay, most important part, and nucleus is present in the cells. Okay, so here I have just given a short detail of all this thing because you know very well that DNA is the part of chromosomes, and chromosomes are found in the nucleus, and nucleus is found in the cells we know very well. Okay, so here cellular DNA is the information source. We know that information means which can transmit from one place to another in a way of source of informations, and DNA exactly work. And for making protein, actually DNA also play an important role in the formation of protein, and also they can transmit message from one place to another. Okay, so it is the most important and beneficial state because today we are going to study only how the characters get expressed and how the characters are generally transferred. We are studying today. Okay, so you all have to make. After that, when the cellular DNA is the information source for making protein, after that DNA make protein in a cell. We know that DNA is found in chromosomes found in nucleus found in cells. So in cell there is a protein formation which is generally done by DNA. Okay, so protein जो भी बनते हैं cells में which is generally made by the DNA. ठीक है, DNA इनको बनाते हैं. After that we know that DNA present in the gene, but all the DNAs are not present in the gene. Okay, several types of small fragments of DNA are generally connected with genes. That are the natures of DNA. It means that you don't get confused that DNA is not found in gene. Okay, DNA small segments are generally attached with genes and they express their character. That's why we are using here DNA present in gene. Okay, only some of their segments are present in gene, not all the DNAs are present in the genes. Okay, most important point. After coming that, we are going to discuss is how the characters are expressed by taking an example of plant. Okay, here we are using an example of plant. हमने यहाँ पे एक plant का example लेके समझते हैं. Suppose tallness of plant, अगर कोई पौधा बड़ा है, suppose अगर कोई पौधा बड़ा है and if the plant is highly tall, अगर वो tall है, so किस base पे वो tall है? We know that the plant hormone is responsible for their tallness. We know that plant hormone is responsible for their tallness. And which type of hormones are generally found in plants? We know that there is several types of hormones like gibberellin hormones, auxins hormones, cytokinin hormone. Several types of hormones are there in a plants. They produce in a specific manner in a plants and they work very well, and by this our plants get tall. Okay, but if they secreted in a less amount, and if we are providing in a less amount to any plant, definitely they result in the responsible for dullness. Okay, that is the most problematic thing for the plant. Suppose we are not providing any specific type of enzymes, hormones to the plants, the plant will not grow. Okay, so that is the important factor. It means their traits are transferring from one place to another, but in a dullness manner. Second step, we have to see that more hormones, more growth. Okay, here we have seen that the plants hormones are totally stopped. यहाँ पे हमने देखा कि जो plant hormones की कमी के कारण here the our plants is totally comes in a dullness stage. When you provide more hormone, like if you are providing in artificial manner like auxin, gibberellin and cytokinin, the plant hormone will grow fast. Okay, more hormone, more growth. जितना hormones देंगे उतना उसकी growth होगी. So most important point that which type of hormones and how they are generally created. Hormones बनते कैसे हैं? Most important point, the enzyme makes the hormones. Important point, कौन बनाते हैं hormones को? The enzyme make the hormone. And enzyme, what is enzyme? Enzyme is a chemical substance which enhance the rate of reaction. Maximum you have to study that term in your chemistry part, not in biology, because enzyme we have to study in 11th class key and lock and key model. Okay, but today I am just going to give you the small definition about the enzyme. Okay, enzyme is a special type of chemical substance which are found inside the human beings and maybe artificially can be accumulated, but they have to enhance the rate of reaction. किसी भी rate of reactions को बढ़ाने के लिए enzyme का use किया जाता है. ठीक है, here enzymes is makes the hormones. It means that here enzyme is totally responsible for making the hormones. So most important point, and that hormones are maybe auxin, okay, and gibberellins, okay, gibberellins and cytokinin, and several type of hormones are responsible for growth of hormones. If you are provide more hormones, definitely the plant will also grow. Next, the DNA genes are responsible for enzyme preparations, and several types of DNA genes makes the enzyme. Okay, we have seen that the hormones are generally created by enzymes, and enzymes are created by DNA, or a gene makes the enzyme. It means that they have the greater opportunity to create such type of enzymes. इसका मतलब DNA जो है gene मिलकर enzymes को बनाते हैं, enzymes जो है hormones को बनाते हैं, and that hormones are responsible for growth of plants. ठीक है, like auxin, gibberellins and cytokinin. ठीक है, but here if there is any alternation in gene, alternation means होता है there is a changes in gene, मतलब उनमें कोई भी अगर कमी हुई या उनकी संख्या में अगर एक्सप्रेशंस आ गए, ठीक है, वो अगर वेरी करते हैं, वेरिएबल्स फॉर्म में होते हैं, किसी में कम किसी में ज्यादा, सो दैट इज कॉल्ड द अल्टरनेशन ऑफ जीन. उनके जीन में किसी चीज की कमी होना या किसी चीज में चेंजेस होना, दैट इज कॉल्ड द अल्टरनेशन. उसी को हम अल्टरनेशन कहते हैं. सो मोस्ट प्रोबेबली द इफेक्ट ऑफ प्लांट विल ऑल्सो doing as a dullness or maybe proper growth. Okay, so that type of alternation are generally done by using several types of enzymes. Okay, now most important point, any alternation in a gene, so that time we have to make a special and make a new enzyme that can work very properly to formation of hormones, and that hormones are generally placed in plants to take a special growth. Okay, so in next process we have to see that if we are going to make an enzyme, definitely the characters of tolerance will be affected. Here we have seen that in a first case the characters of plant is dullness, and here after making a new enzyme the characters of tolerance will be affected. Okay, characters of tolerance will be affected, and after that you will get a new characters. New characters means whether you can get small plant and whether you can get tall plant. So that is how the characters are generally get expressed by using a flowchart. Okay, I have prepared here for flowchart for making you understand that all the topics, how the characters are generally transferred from DNA to only that portions. And here I have taken an example in plant case which type of characters are generally transferring from one parents to another, and after that we got the new result, and that is the produced new character. If you are providing more hormones, then new characters will also a hybrid type, and plants is also in a tall in nature. So this is the adject characters and traits which are generally expressed in. Next we have to discuss the small version of sex determination. Okay, we know that sex determination is a very important part because it play an important role in a human beings that decide whether it is girl or a boy. So we have to study that sex determination play an important role. Okay, why? Because how the sex of newborn individuals are determined. Suppose if there is an infant child are generally get birth. Okay, कोई बच्चा अगर suppose पैदा होता है, how we have to decide whether it is boy and whether it is girl. So for looking their chromosomes in number, first we have to go in a very previous stage and we have to study that in human beings the sex of the individuals is genetically determined. जो भी sex होते हैं वो fetus जो होते हैं वो generally which is already designed in their embryo, ठीक है, before the birth of any child. ये क्या होते हैं, genetically determined हो जाते हैं during the race of their sperm and ovum. Okay, so we will see also by taking an example. After that, in other words, the gene inherited from our parents mean both mother and fathers. Okay, mother and father, both the types of characters are transformed from mothers and fathers to their young ones. Okay, so that is called the sex determinations. And all human beings have 23 pairs of chromosomes. ठीक है, 23 pairs means they have total number of chromosomes is 46. It means they have 44 autosomes and one pair is available for their sex determination. इनके पास whether it is if they have 44 pair of autosomes and it may be XX means it represent girl, and if they have 44 autosomes and XY it represent Y, because here the chromosomes and one pair is available for their sex determination. एक पेयर जो होता है वो उनके sex determination को बताने के लिए होता है. So for different in males and different in females, जो कि मेल में अलग होता है, फीमेल में अलग होता है. Coming to the next important point, they have 44 autosomes in same, but they have one different characters which may be XX in girls and XY in boy. Okay, now taking by using an example, suppose we are going to discuss here we are taking as a boy and here we are taking as a girl. Okay, so boy have different types of chromosomes. Here it is a formation of sperm. Okay, and they contain X and they contain Y. ठीक है, क्योंकि इनमें X होते हैं chromosomes और एक Y होते हैं. And here the gamete की बात करते हैं, they both have X and X. Okay, so apart from that we are going to just see how the sex determination can be identified. So here there is a 2, so we have to take a common. यहाँ पे हम common लेते हैं, इसका मतलब हम X को सिर्फ बाहर ले रहे हैं. And second step we are going to discuss that here you can use 2 because here X is also there and Y is also there. And when you cross between boys to the girls, suppose we are crossing between X and X, the result is girl. And if we are doing that, it means X and Y and this represent boy. Okay, so how it is happened, होता कैसे है ये? When the zygote formation takes place, that time the sperm में race चालू होता है, and that time the sperm which may be X or which may be Y, which reach first in a X chromosomes, that time अगर ये first time यहाँ पे पहुंच गया, इसका मतलब ये XX बना लेते हैं, that is result in the girls. अगर टाइम से पहले रेस में ये पार्टिसिपेट करके ये इनके साथ बन गया, दैट इज मेक्स X एंड Y, क्योंकि X इनके पास ऑलरेडी होता है लेकिन इनके पास X और Y दोनों होते हैं. तो अगर ये रेस में इनसे जीत जाते हैं तो X तो पहले से ही है X और ये X साथ मिलने के साथ यहाँ पे गर्ल्स बना लेगा, और अगर ये Y पहले पहुंच गया तो X और Y के साथ यहाँ पे boy बना लेगा. That is called the sex determination in human beings. Okay, so today we have completed all the hereditary systems in this chapter. In next video we will study the evolutions. Okay, till then whatever I have taught you, you all have to revise very carefully, and if you get any doubt you can ask me. Thank you, students. Good evening class. In previous video we have discussed so many characters of Mendel's experiment, and we have also discussed why Mendel have selected the pea plant only and which types of characters they possess. Okay, and next topic is our the most important and benefits topic for the exam purpose, that is monohybrid cross. Okay, from last video I have told you that what is the meaning of monohybrid. First I want to tell you that mono means single and hybrid means special type of character is called monohybrid. Okay, and here using single trait, इन्होंने एक ही trait को लेकर he has done the experiments of monohybrid cross. Okay, so one by one we have to discuss how the monohybrid cross play an important role. Okay, so he has taken the pure tall, and he has taken a pure drop. Okay, and this is the tall, मतलब ये बड़ा था and this is drop, छोटा था. So pure tall की बात करते हैं, this is called capital T, and drop की बात करते हैं, that is called small t and small t. Okay, so after that they have taken both the types of characters, and this characters is also called parental characters. Okay, this is called P1 or we can say that parental characters means parents के characters थे, and both the characters are homozygous. Okay, wow. Homozygous, what is the meaning of homozygous? Homozygous means same types of characters are called homozygous and different types of characters are called heterozygous. Suppose I am giving an example here. If there is a capital T and one small t, it is a heterozygous. Okay, next, suppose it is a capital T and small r, it is called heterozygous. Here capital T and small y, it is also heterozygous. Homozygous means if both the characters are same, means capital T and small r and capital R and small y. So these all are the characters of homozygous and heterozygous. So Mendel has done the first experiment in monohybrid cross by taking a pure tall and pure drop. Okay, so he has done the experiment of F1 generations. Okay, F1 generation में इन्होंने offspring को देखा. So what was the result? The result was that he has done cross between. Cross between means they have done the cross pollination between first plant to second plant. Okay, suppose here this is the plant. Okay, and this is also a plant. Okay, and this is tall and this is drop. So he has done the cross pollination from that plant to that plant. So same as it is, he has done the cross breeding between same types of characters. It means the character will be capital T and small t. When they concluded both of them, the result capital T and small t. Second stage, they have also done experiment with second character, same as it is, capital T and small t. Okay, next they have done the same experiment. It is called capital T and small t, and next they have done experiment with capital T and small t. The result is also capital T small t. So in F1 generation the result we obtained, and that is all the characters are generally tall. Okay, why? Because here tall is the dominant character and small t is the recessive characters. It means that tall जो है अपने characters को they can show their character in any situation, in anywhere. Okay, so this is tall, and here I am writing here tall is the dominant character and capital T is a recessive characters. Okay, so why? Because capital T is always in capital in nature, because they can able to express their own expression, and here capital T and also small t. So 4 outcomes we have obtained, and all the 4 outcomes are generally tall in nature. There is not a drop. It means the conclusion is that जो parents के जो F1 generation में जो भी daughter cells निकल कर आए, they all are tall, not a drop. सभी के सभी बड़े थे. Okay, then after that they further done experiments with taking 2 characters. Okay, from single trait. Okay, so what was the result? Result क्या था इनका? The result में अगर हम देखेंगे तो most important taking 2 characters, first one and first one. इन्होंने 2 characters को लेकर इन्होंने cross किया. Okay, so the result is most important capital T here and here capital T. Okay, then second he has done capital T with small t, and next he has done with capital cross capital T and small t, and the last one is the strange one. We got that. Okay, what was the strange in F1 generation? This is called F2 generation. Okay, this is filial first generation and this is second generation. Parent generation daughter generation, and second is F2 generations. Okay, this is grandparents, this is father, and this is our sons. Okay, so this is, we can easily note that. The first one is that in F1 generation all the characters are tall. जितने भी characters हमको निकल कर आए F1 generation में, they all are tall. ठीक है, but most important, when we cross breeding between same type of single trait in F2 generation, we got one thing in strange. हमको एक चीज यहाँ पे अलग से मिली, and that is called small. ठीक है, ये small characters हमको निकल कर आया. It means that all these things are tall. ये तीन कैरेक्टर्स जो थे tall निकल कर आए, एक कैरेक्टर जो था वहां पे drop निकल कर आया. It means the changes occurs in F2 generation. F1 generation तक हमें कोई भी changes नहीं मिला. Why? Because all the characters are tall. Because every characters consists capital T. हर एक characters में capital T. This means it is capital T. This is capital T. यहाँ पे भी capital T, यहाँ पे भी capital T है. ठीक है? So this is the tall characters. But after the conclusion of F2 generation, we got that there is a two types of systems are generally occurred. First, this is tall, and second, this is drop. Okay, एक कैरेक्टर से यहाँ पे ड्रॉप निकल कर आया और एक तीन कैरेक्टर्स ऐसे थे जो यहाँ पे टॉल निकल कर आया. It means that the changes of characters may be changes from after the cross breeding when we reaches into the F1 generations. Okay, most important point. F1 generation there is no changes, all the characters are tall. When we reaches to the F2 generation, three characters are tall and one characters are drop. Taking an example of if the parents of mother and father, both the colors are generally black, but when we reaches in a F2 generation, the color of their child may be brown, not black. अगर F1 generation तक अगर माता और पिता के कलर जो है आई कलर जनरली ब्लैक इन कलर है, जरूरी नहीं है कि जो F2 जनरेशन में इन तीनों के ब्लैक होंगे और एक का क्या हो जाएगा, ब्राउन होगा. सो दिस इज द डिफरेंट कैरेक्टर्स. ठीक है, ये जो है आपका डिफरेंट कैरेक्टर्स निकल कर रहा है. सो अपार्ट फ्रॉम दिस वी हैव टू टेक अ स्पेशल लुक ऑफ F2 जनरेशन अकॉर्डिंग टू द जीनोटाइप एंड फिनोटाइप रेशियो. ओके, सो व्हाट इज द जीनोटाइप एंड फिनोटाइप रेशियो? एंड लास्ट I have told you that genotype means the genetic makeup of any organism is called genotypic ratio, and phenotypic means the physical appearances of ratio is called phenotypic ratio. ठीक है, genotypic ratio का मतलब जो genetic characters से related रहते हैं which is concluded as a gene, जो अंदर के characters that is called genotype, and phenotype means the external appearances of characters are called phenotype. So here we have to discuss that most important part of today is genotype and phenotype. So here we have to discuss that what is the exact genotype and phenotype ratio. Okay, so genotype ratio means if you are coming to the next point, there is a different types of characters are there. The first one is tall. It is tall मतलब homozygous. It is homozygous and it is also a homozygous. Why? Because both are consisting of same type of trait. इनमें capital T है, यहाँ पर capital T and capital T है. ठीक है, sorry, small t and small t है. So यहाँ पे अगर कैपिटल टी है और यहाँ पर स्मॉल टी है, मतलब दोनों के कैरेक्टर सेम है. इट मींस दे आर कॉल्ड होमोजाइगस. अगर कमिंग टू द नेक्स्ट पॉइंट, सेकंड एंड थर्ड की बात करते हैं, हियर वन इज कैपिटल एंड सेकंड इज स्मॉल. ठीक है, एक कैपिटल टी है, दूसरा स्मॉल टी है, एक कैपिटल टी है, दूसरा स्मॉल टी है. सो द कंक्लूजन इज दैट द जीनोटाइप रेशियो इज कैपिटल टी, और कैपिटल टी की बात करते हैं तो हियर द जीनोटाइप रेशियो इज 1, एंड एक कैपिटल टी एक स्मॉल टी, एक कैपिटल टी और एक स्मॉल टी, यहाँ पे दो कैरेक्टर्स हैं. इट मींस द सेकंड यहाँ पे आएगा, एंड नेक्स्ट मोस्ट इंपॉर्टेंट एक स्मॉल टी और एक स्मॉल टी. इसका मतलब ये भी इन चारों के आउटकम्स में एक ही बार आया, दैट इज कॉल्ड 1. So genotype ratio is 1:2:1. So most important you have to write down, because most of the time this type of genotype and phenotype ratios are asked in the examinations. Okay, so next, coming to the next important point, we have to discuss that most important point is that after coming to the next point, that we are going to discuss the phenotype ratio. Okay, phenotype ratio is the most important part. Why? Because we are just going to discuss there is a three different types of character. Here is a capital T and here is a capital T and here is a capital T. It means 3 characters are generally tall in nature. यहाँ पे 3 characters जो हैं वो tall को show कर रहा है. It means phenotype ratio may be 3 and 1 is small t and small t. यानी एक character जो है वो drop मतलब small, ठीक है, that is called 1. So most important point, we have completed the monohybrid cross, and monohybrid cross means it is the most important beneficial part. Why? Because monohybrid cross is done by using a single trait. इन्होंने single trait को लेकर cross किया था. The first one is pure tall and second is pure draw, but both the characters are homozygous in nature. जो भी characters से homozygous in nature के थे, they have done the cross breeding between 2 different types of traits by using self pollination, cross pollination की मदद से इन्होंने किया. After F1 generation all the characters are tall. जितने भी characters थे वो सब tall थे, but in F2 generation 3 characters are tall and 1 characters is dropped. एक characters छोटा आया, बाकी 3 characters बड़े आए. So the conclusion is that the genotype ratio is 1:2:1. It means 1 एक यहाँ पे homozygous है, 2 यहाँ पे एक capital T small t 2 characters ऐसे हैं जो heterozygous है और एक जो है homozygous है, बस small t है, ठीक है. That is called genotype ratio. And next is phenotypic ratio में generally 3:1. It means that 3 characters are generally tall. Why? Because 3 characters are consisting, first letter is capital T. यहाँ पे भी T है, यहाँ पे भी T है, यहाँ पे भी T है. So that is called capital T means tall characters. Here small t means drop. So this is enough for monohybrid cross. Next we are going to discuss dihybrid cross. Okay, so most important, next we have to discuss that dihybrid cross. Okay, why in dihybrid cross the Mendel has used 2 different types of traits. Okay, now Mendel is used 2 different types of traits. Why? Because in monohybrid we have seen he has used single trait, and here 2 different types of trait. Capital R, capital Y, and second is small r, and next is capital Y. Okay, so when they found the gamete from both one, they have to make a cross between both capital R and capital Y. में से हमको capital R और capital Y निकल कर आया, and here small r and small y में से when they have crossed between this, the small r and small y, both the types of gametes are generally निकल कर आया, ठीक है. That is called the gametes. इसको हम gametes कहते हैं. After that they had to make a next step of Punnett square. Okay, Punnett square model. Punnett square means this is called the Punnett square because monohybrid cross, and it is. So next here we have to see that we can use by making a Punnett square. Punnett square is the special type of square board in which the monohybrid cross and dihybrid cross can be done. Okay, and this is my homework for you all. You have to make a monohybrid cross in a square method. Okay, and send me in my personal WhatsApp. So here Mendel used 2 different types of traits by doing cross between that, and it is also cross with that, and it is also cross with that, and same. Okay, so after crossing they get the adject form of different types of traits, and traits may be round yellow, traits may be wrinkled green, and traits may be wrinkled yellow and round green. Okay, round green means there is a different types of traits are occurred after crossing between this Punnett square. So I am just drawing and making in one special board, so you all have to follow very strictly. So when they have crossed between capital R, capital Y, the result is also capital R, and capital Y and capital Y. Second, when they cross between that, it means one capital R and one small r, and one capital Y, and second is also capital Y. And when they use, this is also called capital R, capital Y, and here capital Y, small y. Next, they have done cross between capital R, capital Y, and one capital Y and small y. When इसका क्रॉस करने से हमें ये निकल कर आया. Next step, we have done the experiment with this capital R, small r, capital Y. Next, इसके साथ क्रॉस small r, capital Y, ठीक है. Next, they have done the experiment with capital R, small r, and then they have crossed between capital Y and small y. Okay, next step is capital R, one capital Y, and y. So here after that you all have to follow same as it is in first to third square. Next, I am just showing you all have to make in your fair copy. Second, most important is, so all the gametes are generally appeared as, okay, same here, r, small y and small y. Okay, so most important point you have to focus that. After that we are just going to discuss the last one is capital R and small r, capital Y, small y. Next, capital R, capital R, capital Y, small y. Next, capital R, small r, both small y. And next, RR and YY. So this is the contrasting characters of wrinkled green. Okay, so here wrinkled green comes in first time. Round yellow means round yellow where the different types of dominant character, where R is the capital R called dominant or round yellow. जहां जहां पे आपको कैपिटल दिख रहे हैं वो this is called round. ये वाले हैं, ठीक है. This is one also. ऐसे कर करके टोटल आपको 9 characters मिलेंगे. So here the ratio will become as 9:3:3:1. This means 9 round, 1 wrinkled, and 3 wrinkled yellow and 3 round green. Okay, so you have to focus by looking square board, and if you get any doubt you can ask me. And next you have to also focus NCERT book because there is a dihybrid cross is also given. Okay, so here I am just going to describe all the cross between the dihybrid cross. ठीक है, most important if you looking round yellow में here 1 2 3 4 and 5 6 7, okay, 8 and 9. तो ये सब जितने भी कैरेक्टर्स थे, जितने कैपिटल्स थे, that is called round yellow. Wrinkled में देखेंगे तो wrinkled where R is always expressed in a small way like this 1 2 and 3. Here RR short. ओके, तो ये है आपके wrinkled yellow and next remaining is round green, और जितने बचते हैं वो round green हो जाते हैं. So this is the ratio of dihybrid cross. So it is my second homework is that you all have to make a special notes regarding the monohybrid cross in single trait and dihybrid cross in double trait. Okay, and make special notes regarding this by using Punnett square. Punnett square means square board types. Okay, after completing you all have to send me your answer sheet in my personal WhatsApp. Okay, till then whatever I have taught you all have to follow very carefully. Thank you students.